Hi everyone. Tonight's video is an introduction to proteins. So proteins are macromolecules and they are true polymers, which means they are made of repeating subunits called monomers. The monomer in this case is an amino acid. I'm going to show you here a picture of amino acid structure. The backbone of the amino acid is this nitrogen, carbon, carbon right here. So NCC. It's the backbone. It's what we see in every amino acid. Do you see any functional groups in this amino acid? You should recognize an amino group here, and you should recognize a carboxyl group on this end of the amino acid. The carboxyl group can also be called a carboxylic acid. So the name amino acid comes from the two functional groups, the amino and the carboxylic acid. There's also this thing called an R here. What does that R stand for? R stands for an R group. So if we look down here on the left, you can see the example of an amino acid, and you can see this backbone, this NCC. Well, there are 20 different amino acids, and if you look over here in the table, I've highlighted each backbone of each of the 20 different amino acids in yellow. So you can see the amino group, a carbon, and then a carboxyl group. Amino, carbon, carboxyl in each of these amino acids. They have an R group. An R group is what varies between each of the 20 amino acids. So this R group varies from a simple hydrogen in the amino acid glycine to a very complex aromatic unit here in the amino acid tryptophan. You do not need to know all of these different names of the amino acids. You don't need to know the R groups. You should know, however, that there are 20 different amino acids, and how they vary is that their R group is very different. Let's take a closer look at the properties of these different R groups. So some of these R groups have a positive charge, so they act as a base in the acid-base sense of the word. Some of them have a negative charge, and so they function as an acid. Some of them are uncharged, but they're polar, and that makes them hydrophilic or water-loving. That means they dissolve in water. Some of them have special elements or functional groups in the R group, like selenium or a thiol group. These are very polar, so again, they dissolve in water. They are hydrophilic. Some of the R groups are hydrophobic. That means they're nonpolar and they do not dissolve in water. Why do we care so much about the structure of the amino acid monomer and the properties of these R groups? Well, proteins are polymers of 50 to hundreds of amino acids. Little fun side note, if there's less than 50 amino acids, we call them peptides, but you can just call them a small protein. That's fine. So depending on the number and the overall arrangement of amino acids, the entire protein could be hydrophilic. If it's composed of amino acids that all have R groups with hydrophilic properties, then that means the entire protein will dissolve in water. The entire protein could be acidic if it has a lot of amino acids with acidic R groups, or it could be basic or hydrophobic, again, all depending on which amino acids make up this protein. In another way, the protein could have hydrophobic regions or acidic regions, depending on where these amino acids with the different R groups are in the arrangement of amino acids in the protein. So whether a protein is hydrophobic, hydrophilic, acidic, basic, or just has regions with any of these characteristics, it all depends on which amino acids with which R groups a protein is composed of. And looking at specific regions of the protein, it depends on the arrangement or the order of amino acids. This order of amino acids in a protein, which plays such a large part in determining the characteristics of a protein, is referred to as the primary structure of the protein. So how do cells link amino acids together to start to form proteins? They do what's called a dehydration synthesis or condensation reaction. So here in front of you, you see two amino acids. You can see the basic backbone structure of the NCC with an R group and then in our second amino acid, the NCC with an R group. We are going to link these two amino acids together by taking a hydroxyl from the carboxyl of amino acid number 1 and a hydrogen from the amino group of amino acid number 2. This is going to form water, which is where the term dehydration comes from in the dehydration synthesis reaction. And then a new bond called a peptide bond is going to form between the carbon of the carboxyl unit of amino acid 1 and the nitrogen of the amino acid from amino acid 2. What's very important to note is that the peptide bond forms between the carboxyl and the amino of the two amino acids. The R groups are not involved in forming the peptide bond. So in this case, I've put X's over those R groups. I recommend that you do this before you even begin trying to do this reaction. Put an X through each R group so that you are not tempted to use these R groups in forming the peptide bond, because they are not involved in the peptide bond. So you should know that proteins are polymers and that the monomer is an amino acid. You should know the basic structure of an amino acid, that backbone, that NCC. You should know how many amino acids there are. You should know how the amino acids differ from each other. You should know what is an R group and how the R groups differ from each other. And you should know how the number and arrangement of amino acids in a protein impacts its overall structure and properties in terms of its hydrophobic, hydrophilic, or acid or base properties. You should know what we call the order of amino acids in a protein, and you should be able to do a dehydration synthesis reaction between two amino acids to form water and a dipeptide, showing where each product came from in the original reactants. So that's all for tonight.